Bismillahirrahmanirrahim. We will start with gram stain. First, we should place a small drop of bacterial sample on a clean glass slide. We spread it using sterile loop into a thin, even smear. Then we allow it to dry spontaneously in the air. Once dried, we can label the slide properly using a pencil or marker. Then we will go to the next step, fixation. After the smear dries, we fix it either by passing the slide briefly over a flame or using a heater, or fixation by the alcohol. Fixation ensures that the bacteria is to the slide and are not washed away during the staining process. The alternative method for fixation, we can use alcohol. This is 70% alcohol, can be used for fixation in some cases, although heat is a more common method in microbiology lab. What is the gram staining? Gram staining is a differential staining technique used to distinguish gram-positive from gram-negative bacteria. It was introduced by the Danish bacteriologist Christian Gram, and the staining process involves series of steps using 4 solutions. Crystal violet, this is the primary stain. Grams iodine, mordant, alcohol or acetone, decolorizer, and safranin. This is the counter stain. This is the 4 stain in the gram stain. We can go to the step 1. We apply crystal violet to the smear and leave it for 1 minute. This dye penetrates the bacterial cell wall and stain all the cell purple. Then we rinse gently with water. The second step, we add iodine. So we float the slide with grams iodine for 1 minute. Iodine binds with crystal violet to form complex inside the cells. This helps the stain remains trapped in gram-positive bacteria. Then rinse again with water. The third step is decolorization. So now we apply alcohol or acetone for a few seconds. Then we rinse with water immediately to stop the decolorization. The fourth step, safranin, the counter stain for about 1 minute. Safranin stain gram-negative bacteria pink or red. While gram-positive remain purple, the gram-positive purple, the gram-negative it will be red. Rinse and allow the slide to dry. Then we can see the slide under a microscope. We place a drop of immersion oil on the slide and observe it under 100 object. Gram-positive bacteria appear purple. Gram-negative bacteria appear pink or red. So we can say gram staining helps microbiologists identifying the bacteria quickly, which is critical for the diagnosis and antibiotic selection. وبكده نكون وصلنا إلى نهاية our practical today. Thank you.